and we'll get the warm-up taken care of, and then we'll go into the instructions for the lab, and I'll demonstrate to you how you got to do it and all that stuff. The lab today is on the idea of like why cells have to be small to absorb stuff, right? To get stuff into them so they can have nutrients and soak in nutrients and get rid of waste products. And that's what the lab is focused on. So that's why we've got this warm-up today. So the warm-up should be like an old one because we've talked about diffusion and we've talked about osmosis. So hopefully you kind of remember what those things are. So the question is, first, what is diffusion for those of you guys that recall? Kadra?
It's material being spread out from a high concentration to a low concentration until it reaches equilibrium.
Yep.
Why are you doing that?
I just wanted to make sure we got it on video. So Kadra says, did you guys hear what Kadra said?
Not really.
Will you say it again, Kadra? Yeah. And speak directly into the microphone.
That.
She was joking.
Material from a high concentrated area to a low concentrated area until it reaches equilibrium.
Yeah, right. Stuff spreads out from where it's highly concentrated to where it's low concentrated. So when you spray perfume over here, it kind of spreads out equally through the room. Right, we talked about that. We talked about the issues your dad might have in the car with the windows up and the window lock on and stuff like that. Right, all those things are examples of diffusion. Stuff spreads out. How is it a little bit different? Ian, do you remember this, how it's a little bit different than osmosis?
It's different because osmosis is whenever, like, it's in the cell and also stuff comes out rather than...
That was a good, that was close. You're close to having it there. Jack, can you fill in the little part that he missed? Yeah, so it's just going through that membrane is what you're talking about there. So it's the same process that's happening. It's just going through something to make it happen. And the last part of it is how is the cell reliant upon that process of osmosis? Why is that key to a cell? Kadra?
Is it because it allows specific materials on that needs?
Yeah, that's the process, like Kadra said, that allows materials in that the cells need. That's how they get oxygen inside. That's how they get glucose inside. That's how they get rid of the stuff they don't need anymore. But that's the process they use, right? That was what we talked about yesterday when you guys did your skits or your sales pitches about, like, the idea of there's too much stuff on the inside and stuff can't soak in fast enough, and that's what the lab's going to do. Go over the instructions for what this lab does. And we'll go through materials first. One of the first things you need is one of these things. I've cut them into cubes for you, but they're like gelatin cubes. I've cut them into cubes. I can show you how to get them out of here, but it takes like a little scalpel and just kind of get up under them and slide them out of there. But every group needs a cube. It's just gelatin is all it is. Gelatin. And it's got a little chemical indicator in there that'll change color when it comes in contact with specific chemicals. You need yourself a scalpel, and scalpels they were right here. There's a blue little tub. There it is. So scalpels are in here, and as a word of caution, like these things are razor blades with a handle. The time to mess around is not when these things are out, obviously. And please make sure you don't walk off with one of these, because if you're walking around school with one of these, that counts as like a weapon, and that's like the automatic deal. So please make sure you don't accidentally like put one in your pocket. Right? Right. So I'm just saying don't do that. So you need a cube. You're going to need a scalpel. You're going to need a beaker, which is one of these things. You just need one per group. You're going to need one of these. These are a dissecting tray. So yes, stuff has been dissected in here before, but you're going to need one of these just to kind of contain the mess and stuff like that. And then when you're set and ready, you'll come up and you'll get some of this stuff. This is sodium hydroxide. It's not super strong, but still you don't really want to get any on you. It wouldn't be super great if you did. But that's the stuff we got. So with our materials taken care of, I can show you, or I can explain to you what we're going to do. You're going to cut the cube that you get out of here into two different, like, as perfect as you can get them, cubes, right? So, like, squared off cubes. One's going to be larger, that's going to represent a big cell. One's going to be smaller, that's going to represent a smaller cell, a cell that has divided. And what you'll see is there's a difference between how much that sodium hydroxide penetrates into the cell, and that sodium hydroxide is supposed to represent like nutrients and oxygen and stuff like that. In one of the cells, it'll penetrate further in, and most of the cell will get what it needs. In the other cell, it really doesn't penetrate that far. So, procedure say, each group needs a cube from the tray. So to get a cube, you're going to grab yourself a dissecting tray from here, and I can even help you scoop the cubes out, because we don't want them to break before they come out of there. And you want a scalpel, and I'll take them from this end, because I've got it pretty easy on this end. It just goes in. What's that? I know, I'm just scooping one out. I'll scoop these out for you when the time comes. But all I'm doing is scooping one of these things out of here is all I'm doing. So this one's kind of broken up because I've scooped it out before, but you're going to get a cube out of here. Yours won't be broken up like this one. They're all easier to take out after the first one. But you're going to scoop a cube out of here, and this cube, mine's falling apart because it's been cut, but this one, you're going to have like a regular old cube. It's going to go inside your tray, and the next thing you're going to do is you're going to make 2 cubes out of your cube that's there. You're going to use this ruler to do it. And just for reference's sake, right, like we've got an inch side and then a centimeter side, we want to be on the centimeter side. If you're making, the cubes aren't big enough to do it on the inch side, so just don't get it confused here. What you want to do is you want to cut that cube up that you have right there into 2 different size cubes, a small one and a big one. We're going to be pretty specific about how big they have to be. One cube needs to be like 1 by 1 by 1 centimeter. And one cube's going to be double that size. That's going to be 2 by 2 by 2, right? And the way you measure that is you just take your cube, measure 2 centimeters long, make a cut all the way through, measures 2 centimeters wide, and then measure 2 centimeters deep. So you're just cutting this up into like a 2 by 2 by 2 cube, and then into a 1 by 1 by 1 cube. The rest of that stuff, if you cut neatly, you can save just in case something goes wrong. You can reuse it or whatever. But if you do it right, when you've cut your 1 by 1 by 1 and your 2 by 2 by 2, the rest of it is just waste. So you don't really need it unless something goes wrong. So you might hold on to it till the very end, just in case, but otherwise it should be just fine. So you guys with me so far? Yeah. Okay. So the issue you're going to run into a little bit, and this is not what I'd recommend you do because you never cut towards yourself, but you're going to have to, just for demonstration purposes here, is you've got a scalpel, and when you push on this, I don't know if you can see it, but it kind of flexes. So it's almost impossible to get like a perfectly straight line, because you want it to be as perfect as you can. So if you just use like little cuts like that, you don't just get on there and like push it straight down. You just kind of like work it back and forth. You hold this like a pencil and work it back and forth. But you'll cut your cube out, and you want to measure the exact measurements, because rarely do you are you perfect to make it 1 by 1 by 1. So we want to record the exact measurements of like length, width, and height. And you've got a spot to do that right there. So even though you're supposed to make it 2 by 2 by 2, it doesn't always come out exactly 2 by 2 by 2. So give me the actual dimensions of it right there. So far, so good? Yeah. All right. So you'll give me length, width, and height. After that, you'll do the volume of the cube down here. And it says before, because this is before we're going to soak it. So you're going to give me the surface area. The formula for it is right there. Side times number of sides. And then you're going to give me volume, just like it says there. So the formulas for both of those are there, of the small cube and of the large cube. So far, so good? Yeah. Okay. The 2 cubes you cut out are going to go into one of these, and you don't want them touching if you can help it at the bottom, because if they touch, they kind of seal together, and then the liquid can't touch them. So you want them like both in the bottom, and you want them like separate a little so that they can like, the sodium hydroxide can surround them. You know what I'm saying? Okay, so you put 2 in one, and then you'll come up, and I'm going to take that sodium hydroxide and pour just enough in your beaker to cover over the top so they're all the way covered. Then we can flip the pages. So on the second page, finishing off the instructions here, what you're going to do is you're going to wait 7 minutes, and what you do in those 7 minutes is up to you. You can do the calculations for surface area. You might be able to look at some of the questions and figure stuff out. But you got 7 minutes where you're just going to start a timer and let this stuff soak. After 7 minutes, the sodium hydroxide, you can just put it in the sink with some water behind it, and it'll be fine. You can just run some water behind it for a little while. Then remove the cubes. So you're going to kind of pour the liquid out, right, without dumping the cubes out if you can. Then you're going to take your cubes and they're going to go back in your tray. And what it's going to look like is they will have changed color. As deep as this liquid like penetrated into them or diffused into them, any place that liquid touches, it'll change color. And I want you to cut off all the stuff that changed color, because that represents like the stuff where the nutrients got, and that stuff would still be alive. What we're trying to do is figure out what's the surface area and volume of the thing that's left behind where nutrients wouldn't have got to, if that makes sense, like the part of the cell that would have died. So you'll cut off all the stuff where it changed color. You'll be able to see it change color instantly as soon as the stuff hits there. Cut away the stuff that's left, then you're going to measure length, width, and height again. You're going to calculate surface area and volume again. And then you've got a little grid to fill out down here, like how big was the surface area before, how big was the surface area after, what volume change. So it's just asking you to compare like the before you soaked it to the after you soaked it and cut it up. Then you got some questions to answer, and that's the end. So that's the plan, that's the lab for today.